The study that I'm presenting is a prospective single-center cohort study to evaluate the effect of the gut microbiome at baseline to response to immunotherapy among Indian patients with lung cancer. So the background of the study is that we all know that the lung cancer burden in India is high, and most lung cancer in India is still tobacco associated. And when we look at the usage of immune checkpoint inhibitors in Indian patients, there is some data to actually say that only 1% of all eligible patients for immunotherapy actually end up taking immunotherapy in India. So there is the largest cohort of around 1,000 patients, which DMH was included, which actually looked at immunotherapy usage in India. Most immunotherapy usage was in second or third line. The median number of cycles was 5, and most patients took it for a short course, which was only around 3 months. And 1/4 of the patients actually stopped treatment despite the short course because of resource constraints. So now there is also emerging data to look at alternative dosing of immunotherapy. We're looking at low dose. We know that there are certain trials going on looking at low dose, fixed dose regimens, and whether you can increase the interval between doses. So the need of the hour for Indian patients eligible to take immunotherapy, which is actually quite a large number, is that do we have a foolproof or predictive biomarker to predict who will respond to immunotherapy? PD-L1 we do have, it is accessible. Even in the 1,000 patients cohort, only around 7% of patients actually underwent PD-L1 testing, maybe because mostly it was in second and third line. MSI TMB not really accessible to anybody else. So, and is there a way to predict responders to these alternative dosing regimens, which would actually have a huge financial implication in our setting? And because anyway, we have now real-world evidence which shows that most patients are on short-course immunotherapy anyway. So the rationale behind the use of gut microbiome as a potential predictive biomarker is that we do have data initially from melanoma and RCC, which shows that gut microbiome can affect responses to immunotherapy. In fact, people are doing fecal microbiome transplants. Now, in lung cancer, there are very limited data which mostly look at non-small cell lung cancer. There are 4 small cohorts, the largest cohort being around 70 patients from Japan, which actually shows that a higher overall bacterial diversity is measured by something called the Shannon index affects the disease response. And you can predict immune responders versus non-responders. And also presence or absence of certain species can direct the way they respond to immunotherapy. So for example, the figure on the right, certain bacteria have a good response, have a positive effect on immunotherapy response. Certain bacteria if present are predictive of not a good response. So often these responses are presented in something called the Shannon index in histogram form. Presence of particular species, we often look at what is called heat maps of individual samples, and then presence or absence of particular species can also be looked at in histograms. So our research question is that does the gut microbiome affect response to immunotherapy among Indian patients with lung cancer on receiving any form of immunotherapy? And the hypothesis is that the gut microbiome diversity as measured by the Shannon index helps predict response to immunotherapy. So our primary objective is to actually measure this thing called the alpha diversity of the gut microbiome and whether it can predict responders versus non-responders. Moving on to our secondary objective, we also want to look at the subgroup of patients who get an alternative dose of immunotherapy, which essentially could mean whether they got low dose, whether they got a short course, whether the interval between immunotherapy administration was increased, or whether they had a fixed dose, whether this alpha diversity in this subgroup of patients, whether it predicted for disease response. And then the other secondary endpoint is to look at, is there a gut microbiome signature? Now, in terms of a signature, this is essentially what I showed you earlier. So we would try and want to do a heat map of individual samples and to looking at individual samples and species of bacteria at baseline, and to see if there is a relative abundance of any particular bacteria between responders and non-responders. We would also want to look at the differential response rates and PFS and OS among patients who demonstrate either a high alpha diversity versus a low diversity, and also based on the relative abundance of specific bacteria. And also there is some data to look at correlation between gut microbiome and immune-related toxicity. Want to look at the longitudinal change in the gut microbiome signature, so we will have a serial sample, and also the differences between our patients and their Western counterparts. So this is a prospective single-center observation cohort done at CMC Vellore. The study duration will be two years, and follow-up will be for 2 more years. Sample size, there is hardly any prior data. So this is going to be like a pilot or proof of concept study with in terms of feasibility. We did analyze last 3 years of patients with lung cancer who got any form of at least one dose of immunotherapy. We had 80 patients over the last 3 years, predominantly low dose immunotherapy. So we will include adult patients starting treatment with immunotherapy for lung cancer who will not have obviously had any prior immunotherapy exposure. We will exclude pediatric and hemoc patients and patients with pre-existing autoimmune conditions and also patients with the intestinal stroma. Now, this gut microbiome testing, how is it done? This is the lab part of the research. So we will collect a stool sample, preferably between 6:30 to 8:30 a.m., preferably in hospital. If not, it has to reach the lab within 2 hours and immediately it will be frozen. And the method we will use is something called a 16S RNA sequencing. So essentially, this picture shows we will extract the DNA and do this RNA sequencing. And the results we get what you see on the right. So we get this in terms of we get the diversity. We get the relative abundance. This is the heat map. We also get a phylogenetic a taxonomy tree, and we will then draw these box plots. So this is our study schema. Now, all the sample right at the bottom. So other than what is standard of care for each of these patients, we're only going to take an extra stool sample. So this stool sample will be done at baseline and at the first response assessment at 3 to 4 months. Now, the reason for this is that as for that retrospective, the real world evidence cohort, most patients got immunotherapy for 3 months, and most patients have their first disease response at 3 months. We are not really expecting majority of our patients on this alternative dose regimens to be taking it beyond that. If they take it beyond that, well and good. So we expect we will do a second sample at 3 to 4 months, and of course at progression, and also if and when there is a grade 3 or grade 4 immune adverse event. And to the right, that is my study. Now, in terms of budget and funding,
1 minute left, ma'am.
Our microbiology department has given us a cost of 8,000 per sample. So the budget comes to around 12 lakhs, and I can get around 3 lakhs internal funding. So this is the proposed timeline for the study. If possible, I'll try and get IC approval within the next 3 months, and hopefully recruit over the next 2 years, and by 2026 be done with it. And so, in terms of our primary endpoint, this is our analysis plan. And we will analyze the alpha diversity by comparing the Shannon index between responders and non-responders. For this, actually, we will have to do an ROC curve and to determine what is a significant threshold to call what is a high diversity and what is a low diversity, and then compare this between responders and non-responders. We'll also look at PFS movers in patients with high and low alpha diversity. Now, how is this different from other gut microbiome studies? Well, the technique, ideally what is done is the metagenomics, that is more, but however, a 16-hour RNA is more feasible. It is likely to yield an answer than a pilot study like this, which is going to be proof of concept, and then maybe we can take it forward in other diseases and other cohort. Population, well, this is novel because there is no data, especially in Indian setting and let alone in an alternative setting. Are we addressing a knowledge gap? I think so, because there is a clinical need to identify patients who will respond to immunotherapy, and of course, there are tremendous financial implications. And we hope to identify Indian patients with lung cancer who will benefit from this and the alternative setting. Now, I just came to Aspire with what is on the left. I just brought it as a basket study, thinking I will evaluate this in all Indian patients with who take immunotherapy. What I realized on day one is that I hadn't really thought about heterogeneity, gut cancers versus non-gut cancers. And then I was going on talking about gut microbiome, but I'm not a basic science researcher. I'm only a medical oncologist, and I didn't know what to do. So I only to have my mentors kind of guide me. Going back with something which is resembling a fairly okay protocol document, but I will work on this. So these are my acknowledgements. Thank you for your time.